as the traditional custodians of the various lands upon which we are all meeting today, and the areas from which our patients are spiritually connected. We are hosting this webinar from the traditional lands of the Gadigal and Bidjigal people of the Eora Nation in the east, and the Burramudjigal people of the Darug Nation in the west of Sydney. We acknowledge the strength, wisdom, compassion, and care Aboriginal people have for their kinship, language, culture, and spiritual connection to country. We pay our respects to the elders, family members, and children who are our future leaders. We also acknowledge our community members, our Aboriginal staff, and the Aboriginal services and organisations who work closely with us to improve the health and well-being of children, Aboriginal children, and young people, their families and communities. So once again, welcome to our Advanced Therapeutic Webinar series, today focusing on drug discovery and adaptive clinical trials. Firstly, some housekeeping. A video of the webinar will be emailed to all attendees and available via our website in the coming days. Our Q&A panel will be answering selected questions at the end of the webinar, so please submit questions throughout the webinar via the Q&A tab in the Zoom controls. If you see a question submitted that you would also like to have answered, you can upvote the question by clicking on the thumbs up image beside it. We're also providing answers to any unanswered questions via our website or email. So today we're very fortunate to have two speakers who are leaders in their respective fields of drug discovery pathways and adaptive clinical trial design, Dr. Ian Street and Professor Tom Snelling. Over his 31-year career, Dr. Street has worked in industry and academia in the United States, Canada, and Australia, dedicating his career to bridging the gap between a great idea and the technical and commercial realities of translating that idea into a new medicine. He has established several collaborative platforms to facilitate research translation, including the WeHi High Throughput Chemical Screening Laboratory, the first academic group of its kind in Australia, and the Cancer Therapeutics Cooperative Research Centre, Australia's first cooperative enterprise dedicated to the development of new highly differentiated small molecule cancer therapies. Ian is a Director of Therapeutics Innovation for Kids, a platform designed to discover, development and commercialise novel therapies tailored to treat childhood cancer. Therapeutic Innovations for Kids is part of the Children's Cancer Institute, which is based in Randwick, Sydney. Professor Snelling is the Director of Health and Clinical Analytics in the School of Public Health, University of Sydney, and is an infectious diseases physician at Sydney Children's Hospital Network. He instigated and now heads the network's new Learning Health initiative, thanks to a major philanthropic commitment from the Sydney Children's Hospital Foundation. Tom is pioneering the application of novel approaches in design, coordination, implementation, and analysis of public interest studies. He successfully leads a number of multi-institutional collaborative learning health projects across Australia. These include Bayesian adaptive studies such as improving treatment and prevention of severe gastroenteritis in remote Aboriginal children, primary prevention of food allergies in children, and improving timelines for routine immunisation. I welcome Dr. Street, who will start this webinar with an overview of drug discovery. Thank you, Ian.
Thanks, Craig. I'll just share my screen. Okay, thanks. So I guess my task in the next 15 minutes is really to give you a brief overview into the process of drug discovery. So as Craig mentioned at CCI, we're currently in the process of setting up an end-to-end paediatric drug discovery pipeline, which is basically based around the knowledge and data that's been gathered in from the zero clinical trial, and establishing a computational target identification group and a functional biology group for target validation to develop new drugs to treat pediatric illnesses that have an unmet medical need. So, as Craig mentioned, also the drug discovery arm of this endeavor is called Therapeutic Innovations for Kids. So, what is drug discovery? Well, it's really the first stage in the drug development pipeline, which I'm sure many of you know about the latter stages of this, the clinical trials phase. But before we get to that stage, we really have to turn the idea, which has come from around the biology and how you might use that to treat a particular illness in children or adults, and convert that into an actual chemical entity, a new investigational drug, which will be used to essentially test that hypothesis in clinical trials. So typically, the discovery process is divided into three stages. The first one is the hit identification, and this is really where you first introduce chemistry to the biology of the target and find simple molecules that will form the starting place for your medicinal chemistry program. Lead generation is really the next phase of the operation, and this is where chemistry begins in earnest. And this stage is really about triaging the chemistry that comes out of the first stage to find those chemical scaffolds that have the best development potential. Then the last stage really in this process is called lead optimization. And this is where you try and bring everything together, both the chemical and the biology together into a single molecule, which forms the investigational drug, which will go into clinical trials, known as a clinical candidate. So, although we're only just starting work on our first projects in Think, so what I'm going to do today is use a project that we ran in the Cancer Therapeutics CRC to illustrate the process. And this is a project that came to the CRC from Steve Jane and David Curtis at Monash around a protein called protein arginine methyltransferase 5, or PRMT5. So I could probably easily spend a 45-minute seminar just talking about the biology of this target. Is really fascinating. But needless to say, for the purposes of this talk, all we need to know is that Steve and David have presented a really compelling case why we should tackle this project to treat potentially AML and myelodysplastic syndrome. So we took this on in 2011. Within the CRC, we conducted the drug discovery process over about 5 years to 2016, and then licensed the whole package to Merck Sharp and Dohme in the US to take into clinical trials. So I'm just going to take you through this process as a quick introduction. So PRMT5 is actually a methyltransferase, as the name implies. Essentially, it's an enzyme which methylates specific arginine residues on target proteins. And this methylation can have a profound effect on the activity of the protein and how different types of complexes are assembled around the protein. So the first step when a project comes to us is if the biology and therapeutic hypothesis get a tick, then we have a look at the the protein with a view to its druggability. So this is probably a word you've heard many times. Is this a druggable target? Well, PRMT5 has basically got one big thing going for it, in that it's an enzyme and naturally uses a small molecule binding site. A small molecule is part of its catalytic machinery, and therefore has evolved a nice small molecule pocket to potentially bind a drug. But in our analysis, essentially, we also found that the groove here, down here towards the bottom of the structure, where the peptide, arginine peptide, binds into the active site, was also likely to be a reasonable drug discovery, drug binding pocket. So this protein, in fact, got a big tick from a couple of points of view in terms of its druggability. The next step in launching a drug discovery program is the design phase. And this really starts at the end of the process, where you want to get to. So based on the biology, you ask a number of questions. What diseases are we going to treat? What patient populations are likely to benefit? And how are we going to deliver this drug in the clinic? And from that, you put together what we call a target product profile. And this is essentially, in that happy day, you might get regulatory approval for this drug and launch it into use in the clinic. This might be the label that goes on the package. From this TPP, you can work backwards and design a molecule that the parameters, the properties of a molecule that you would need to fulfill that target product profile. And then based on that candidate profile, you work backwards down the screening cascade and work out profiles for the simplified chemistry that you might identify at each stage, all the way back to the start of the project. And by doing this, this really gives you a fairly rigorous framework of boxes that you can tick in terms of the molecules that you produce to decide whether you want to progress them down the pipeline and invest more resources in developing them. So for PRMT5, this was to treat AML or MDS. And we know there's specific mutations which make these diseases both very sensitive to treatment with PRMT5 inhibitors. Unfortunately, I can't disclose these at the moment because the patent hasn't yet been published. We wanted to go oral dosing once or twice a day at maximum, and this was to be used as a single agent or in combination with standard of care chemistry and this therapy. And this really determines the types of models that you would use in terms of safety and efficacy towards the end of the program. So the first step, having set our design parameters, were really to find the starting place for the chemistry. And there are a number of different approaches that you can use: high throughput screening, fragment screening, structure-based design. But because this is a completely novel target, when we started the project, essentially we went for the big guns, high throughput screening. So essentially we produced bucket loads of recombinant PRMT5, set up a high capacity assay, and launched the screening campaign, screening about 350,000 compounds. And to cut a long story short, we went through the process of triaging all the hits, and we ended up prioritizing two different chemical series, two different chemical scaffolds, if you like, to go into lead generation. So these were interested not only because of their biological, their biochemical activity against the target, but essentially we found out through a series of mechanistic studies that one series bound in the SAM pocket and the other series, series 2, actually bound in the peptide binding domain. And this gave us another way of mitigating the risk across this target, in that we could tackle the approach to 2 different sites on the enzyme. So these move into lead generation, and essentially lead generation is the process, as I mentioned, where you look at the hits, the chemistry that's coming from the hit discovery, and start to explore this chemistry around the chemical scaffold. And as well as looking at the variations that are made, an effect on the potency of against the target, you look at also the effects that you have on other properties of the molecule that you're going to need to develop a good drug, like the pharmacokinetics, the metabolism, how these changes affect those properties, if there are any particular targets that you don't want to hit, like cardiac channels, whether the changes in structure effect induce any of these off-target activities which are undesirable. And essentially you work through a number of iterations of the chemistry to get a good view of how these things will change across the scaffold. Key to this is the screening cascade. And this is where really a depth of knowledge about the biology that you're targeting really comes into play. Starts with very simple assays. Does this compound hit the target biochemically? Does it get into cells and inhibit the target there in the cell? What other activities has it got? Does it have basic exposure in rodent models, and the off-target activities? And so this is goes round and round until essentially we've explored both series and came to a conclusion. So this is just one of the things that you really look for in this part of so this is a series 2 compound. Essentially, you can see on the left, that blue circle, the chemists found that they could vary the structure around this part of the molecule really quite drastically without affecting the potency. And this is great because it gave them somewhere to moderate, effects like solubility by changing the chemistry there. On the right-hand side was this tetrahydroisoquinoline, which basically you couldn't really modify at all without dramatically losing the potency. And this became something we had to tackle later on in the project, because this is a potential safety liability, plus also it turned into an IP liability later in the project. So at the end of the lead generation, you end up with like a score sheet based on the criteria that you've set at the start of the project. Red for rubbish, green for great, orange for maybe. So as you can see from this score sheet, essentially the series 2 got the Guernsey to go into lead optimization, and we abandoned series 1 at this point in the process. So lead optimization, I guess it's also important to realize at this point of the program, you don't have a molecule that has all the desirable criteria that you have, but rather a series of molecules, some of which have good properties and some of which have some bad properties, but there's not an ideal molecule at this time. So lead optimization is about baking all those good properties into a single molecule. And essentially it's the same process. The medicinal chemists design molecules. They go into the screening cascade. You start by asking simple questions first. What are you hitting the target? What else are you hitting? Does it matter? Is it likely to cause safety issues? then progressed down to sort of more complex matters like the pharmacokinetics, the metabolism, the distribution, and address those parameters. And every time you design a new molecule, essentially you work your way around the cycle, down the screening cascade until you basically hit a no, this is not good enough. Then you go back and design a new molecule to address that issue and hopefully work a bit further down around the cycle until eventually you've completed and you've got a molecule that meets all the criteria that you've specified in your target candidate profile. So this is the complete chemistry story for this amino-indane series that we developed, starting from the hit to the final molecule, 293, which was one of the molecules that Merck licensed. And as you can see, it looks really, really simple when you put it all on one slide like this. But during the course of this project, we made probably nearly 1,500 new compounds to get from there to there. So this molecule is one of the key value drivers for commercialization of a project like PRMT5, because, you know, if a big pharma licenses this program, this saves them probably 5 years of work and they can basically start from this molecule. So this has to be novel. It has to be protected in terms of the intellectual property. And around this project, we filed, I think it was 10 composition of matter patents to get by the time we'd gotten to this point. So the next question is preclinical proof of concept. And these are just some of the results that David Curtis and Emma Toulmin, who worked in a postdoc in his lab, ran some of the more complex proof of concept preclinical models. This is a syngeneic AML model, mouse model. And the question, so these were treated for a short time with a PRMT5 inhibitor, 14 days after disease had established. And then basically we looked at overall survival. And you see, certainly in the P53 wild type mice, there was a considerable increase in overall survival, survival benefit there for those mice. If you look at the bone marrow, essentially you can see the untreated mice, the bone marrow is just full of these blasts containing these dimethylated arginine proteins and more or less cleared, and essentially we restored the bone marrow function after treatment. So it's this type of information that really makes a compelling case to a pharma or biotech company that they want to take this project on. So one of the things I really enjoy about working in drug discovery is that it is a team sport. You need to work with a lot of different experts, a lot of people with different knowledge, you know, both scientific as well as business, the original biologists, of course, clinicians who might want to guide the way that you develop the preclinical package, and of course, manage the intellectual property. So this is, I think, a fantastic part of drug discovery. And then just to wrap up then, basically I want to, of course, acknowledge my, you know, the people that also worked on this project. As you can see, there's a long list from the Cancer Therapeutics and also my colleagues at CCI, some of which were part of this original project, but are now working to set up Think. And that's the introduction to drug discovery. And I'll hand over to Tom, if I can find out how to do that. There we go.
Okay, so I'm going to give a very brief overview, introduction into adaptive clinical trials. Most of my work is really in the later phase, comparative effectiveness like studies, but a lot of the methods that we use really have their origins in early phase clinical trials. Really, there have been two big philosophical developments in clinical trials over the past decade. One has been a move to platforms or master protocols. The idea here is that rather than continuing to set up clinical trials as standalone experiments, that we should have a more organized and coordinated approach where if we've got a particular clinical problem, clinical disease or an indication, that rather than set up multiple clinical trials for each candidate drug, that we should have a standardized protocol and a standardized approach to evaluating those drug candidates. So that's this idea of master protocols, and this has really been championed by a number of groups, including from the FDA itself. And then the other area of development has been in moving away from traditional fixed design clinical trials to adaptive trials. So this is the idea that things will happen in the course of a trial. You're collecting information, you're collecting data. It doesn't make a lot of sense to not be able to change elements of your trial as you go. You should be able to use the information that you're collecting and that you're accumulating in order to optimize the design of your trial. And so that's this idea of trials that are adaptive. And the important thing there is that those adaptations that you make need to be pre-specified. So this is not making decisions and ad hoc decisions as you go. This is designing your trial upfront in a way that you have specific pre-specified rules that allow you to make changes as you go. And what I'm talking about today mostly is the intersection between those two ideas, and that sort of culminates in the idea of adaptive platform trials. So this is a fairly recent review of master protocol designed trials from a couple of years ago, showing that really these have started to become of interest and be designed and implemented over the last 10 years or so. There are three sort of major flavors of these platform designs. One is the basket trial, and most of these trials have been sort of basket designs. Another is the umbrella design, and thirdly, the full platform design. So a basket trial, the idea, really the setup for a basket trial is where you have a specific drug. So you have a single drug or a drug candidate, and really you're trying to find a particular patient group in whom that drug will be effective. And so that might be, for example, a drug that targets a specific mutation in cancer, and that mutation might be present across a range of different cancer types. And the traditional approach would be to set up trials in each of those cancer types and hope that you identify a particular cancer type that your drug candidate works in. The idea behind a basket trial is rather than having separate trials, you have effectively a single trial, and you are evaluating your candidate in all of those patient types who share something in common, generally a common mutation in the case of cancer trials. So there have been about 50 of these basket trial designs that have been registered. Almost all of them have been in oncology. They're almost all, as you can imagine, early phase, so phase 1 and phase 2 or both. They're generally open label designed. They generally don't have a control group, and they're not randomized. So you might have, for example, a target response rate for each of the patient types, each of the cancer types. And you effectively keep enrolling participants to that patient type until you hit some pre-specified criteria of success, usually defined by hitting a high probability of meeting the minimum response rate and having an acceptable toxicity profile. So these basket trials are bigger than sort of traditional phase 1, phase 2 designs. They're generally sort of several hundred participants, reflecting the breadth of different patient types that you might have in a basket, but it scales so that rather than having dozens or a couple hundred patients per patient type, you potentially can leverage information across the different patient types. So you need a smaller number of patients overall to identify the patient types that are most promising. And so the other setup for a master protocol trial is the idea of an umbrella trial, which is the opposite in a way. Rather than having a single drug with multiple disease targets, you've coming at from the perspective of a single disease, and you've got multiple potential drug candidates, and you want to either evaluate each of those candidates against each other or against the standard of care, or you might have different patient subtypes with that specific disease, with that particular cancer, for example, or whatever disease is of interest, and you might target those different candidates drugs to the specific subtypes. And so there are not a lot of umbrella trials that have actually been implemented. There are a couple dozen of these umbrella trials now, again, almost exclusively in the oncology space. And again, generally implemented as early phase trials, either phase 1 or phase 2, or a bit of phase 1 and phase 2. Again, these are almost always open label. In the umbrella design, because you do have the ability to directly compare different candidates against each other for a specific patient type or disease, they're more often randomized than in the basket trial design, and typically umbrella trials have evaluated several different drug candidates simultaneously, or sometimes they can be compared and assessed consecutively. And again, typically umbrella designs enroll several hundred participants. So the next step from the umbrella and the basket design is the full platform trial design. And I think of these as being a combination of the basket and the umbrella. So the basket was one drug, multiple disease or patient types. The umbrella was one patient type, but potentially multiple drug candidates, whereas the platform basically does both. You might have multiple types of patient, but you also have multiple drugs, and you want to identify either any of the drug candidates that are most promising, or any that are likely to be better than the standard of care. Or you might want to directly compare those candidates against each other, with the idea that you really just want to take forward into a definitive trial the most promising drug for that specific patient type. The key feature of the platform trial is the idea that you perform consecutive analyses. So rather than just enrolling and having a single analysis at the end, you're actually analysing the data as you go. And in this diagram, they only depict 3 analyses, but in fact, the way that we implement these designs, you might have dozens of these analyses as the platform is being implemented. And what that allows you to do is to be able to drop any treatments that are not appearing promising. So treatments that have a low probability of being effective or of being the best treatment or intervention for that particular patient type. But it also allows you to introduce new interventions, new treatments over time. So if a new candidate becomes available for a specific patient type, in theory, those new interventions can just be added into the platform. So increasingly, people are thinking about these platforms as being almost perpetual designs where you effectively continue enrolling into the platform as long as you have a candidate to evaluate. And there are, as of about 18 months ago, there were about 20 of these platform trials that had been registered and being implemented, again, mostly in the oncology space, but not exclusively. The platform trial designs, again, often early phase, but more commonly sort of phase 2 or phase 3, or a seamless phase 2 and 3. And since COVID, there's been a small explosion in these platform trials, and we're involved in a couple of these, looking at repurposing drugs for use for COVID-19. So the ASCOT ADAPT trial, which is an Australian platform trial that we're designed and we're involved in the implementation of, is looking at various licensed drugs for COVID-19 treatment, and REMAP CAP or REMAP COVID is another international platform trial that's also looking at repurposing various antiviral and immune modulating therapies within a single platform. And so these platform trials tend to be larger trials and enroll large numbers of participants, and actually enrolling several thousand or more participants is not uncommon now for these platform designs. Because the flavor of this seminar is around drug discovery, I thought I might say something also about Bayesian N of 1 trial designs. So this is, while people have talked about N of 1 designs quite a lot, there hasn't been a lot of implementation of these sort of Bayesian N of 1 designs, but they are appealing for situations where you've got a diverse patient population. You've got some people who with a particular disease who are likely to respond and some who aren't, and you really want to identify which patients, which individuals, I should say, are likely to benefit from a specific targeted therapy and who isn't. And so the idea of an N of 1 design is that you randomize, everyone effectively receives the treatment, and typically they would receive a matched placebo as well, and they're effectively alternating on and off the treatment and the placebo over time, where the order in which they receive treatment and placebo is randomized. And ideally, they don't know whether they're receiving the active drug or the placebo. You have some outcome that you're trying to assess, and you assess that in each of these treatment periods. And you're really looking for to identify for each individual whether their outcomes are better while they're on the drug compared to when they're on the placebo. And when you do that within a Bayesian context, because it's adaptive, you can essentially continue to alternate people on and off treatment until you can be certain either that the treatment is beneficial, or be certain that the treatment's unlikely to be beneficial for that patient. And so, and it depends on the condition, obviously, but it wouldn't be uncommon to be able to identify a responder after several alternations on and off treatment. So this is a design that we've recently worked on with Adam Jaffe at Randwick and Peter Walk in Newcastle for assessing individual responses to disease-modifying drugs for cystic fibrosis for people with rare mutations who don't currently have a licensed molecular therapy. And the proposal is to alternate these patients on and off a particular molecular therapy to hopefully identify who are those who benefit and who should ideally continue on that treatment, and who are the individuals who are not responders and shouldn't continue on treatment that is obviously expensive and could be toxic. So we think that that's a potentially exciting way of the future. I might just skip forward to this slide, which is just a summary of which design is best. If you've got a drug and you want to find a disease or a target for your specific drug, then that lends itself to a basket design. If you've got a disease and you want to find a drug for a specific disease, then that lends itself to the umbrella design. If you actually want to identify any promising or the most promising drug against a disease at the lowest costs and the most efficient way possible, then an adaptive platform design is best. And if you want to assign treatment to participants in a way that maximizes their outcomes, then again, a Bayesian platform trial is the most suitable option, and that's what we're doing with the BEAT-CF platform trial at the moment. So not so much a question of identifying a treatment that works, but trying to optimize the outcomes for people who are participating in the study. If you want to really personalize treatment and you've got a drug or a compound that has relatively short-term effects that wash out relatively quickly, then we think that these Bayesian N of 1 designs are most promising. There are a number of challenges, not just statistical, but also with funding these platform designs. There are challenges with implementation and how you report the outcomes or the results of a study that is effectively ongoing. And if you're interested to find out more, we've developed a number of resources at the Adaptive Health Intelligence website. And so I will stop and hand back to you, Craig.
Thank you very much. So thank you, Ian and Tom, for wonderful presentations, and thank you for those who have submitted questions. I would encourage the audience to continue to submit questions via the Q&A tab. It's now my pleasure to introduce Professor Chris Cowell, who is the Director of Research at Sydney Children's Hospital Network, and Dr. Laura Fawcett, who's a respiratory consultant and advanced therapeutic lead at Sydney Children's Hospital, Randwick, who will be facilitating our question and answer session. So thank you, Chris and Laura.
Okay, so I have a question for Ian about the THINK initiative, and it's a great question because really can THINK be extended to disorders outside of cancer, or is it a model that could be replicated certainly for other disease groupings? Need to take yourself off mute, Ian.
Yes, absolutely. And we're already engaged in a number of discussions through Luminesce to basically look at other diseases outside of cancer as well and take those on. So yeah, it's a model that is flexible. The chemistry, the process is therapeutic area agnostic. All we need is the biology and the target, and of course the funding to run the projects.
Thank you, Ian. I've got a question here for Tom. So Lisa Melton has asked, in the basket trial model, something that is gaining traction, is this something that is gaining traction with regulators? So she's thinking about rare disease trials and trials without placebo arms.
Yeah, I think I mean, most of the work in these adaptive designs has really been in the device regulation space, and they've certainly for the past 10 or more years have been very supportive of regulation and licensure of devices using these sorts of designs. Certainly in the last few years, even in the drug development space, the FDA and the EMA have taken a much more supportive approach to accepting evidence from these sorts of Bayesian adaptive designs to support licensure of new drugs. And as you can imagine, especially a basket design where a sponsor has got a candidate that could be effective across a range of cancers, it just makes much more sense to implement as a basket design rather than multiple parallel designs. One of the efficiencies in the basket design is that you can effectively share information across different cancers. So cancers that share a particular mutation, but they're different cancers. And the challenge for the regulators has been around how much borrowing and sharing of information are they willing to accept across different cancer types. And so rather than assuming that every cancer is completely different, and that you can't learn anything about response to a drug from one cancer type to another within the basket design framework, where you have a hierarchical model that allows you to share information. It's always been a bit of a uncertainty about exactly how much sharing to allow. But I think as we're getting more and more to precision therapies, where every cancer or every disease type is relatively rare, I think regulators are realizing actually this is a much better approach than not having any data at all.
I have a question for Ian, and you didn't mention it, but it sort of was implicit in your presentation that there probably is quite a high attrition rate in the drug discovery program that you outlaid. And so it's a question partly, why is that? And then where do you overlay the IP protection, given that you're dealing with that element as well?
Yeah, I mean, there is a higher attrition rate. I think in the discovery phase, it's mainly, well, there are two principal causes, obviously. Either we can't make a molecule which will fit all our criteria in terms of where we want to get to, usually because of the target, the biology of the target. And the other one is that basically we make a good molecule, but the biology doesn't hang together. And so usually that one's not a full stop, because although you're not maybe doing an AML project anymore, you've still got the investment in chemistry is still there. And we've done a number of projects where we've stopped working on a project when the biology, the original hypothesis didn't hold water. But then a year or so later, somebody's published a new indication. And then we've gone back, reactivated the program. So it's a combination of that, the druggability, the ability to make a good drug around a particular biology and the biology. So in terms of the IP, this is also another complicated question. But generally, you hold off as long as you can before patenting chemistry. And this is largely because the fixed lifespan. So once you file a patent application, that 20-year protection starts ticking. So obviously, if you file very early, by the time you get to actually licensing that, you've lost several years of your patent protection. But also, a patent is really only effective, at its most effective, when you've captured the actual structure of the clinical molecule in a patent. If you patent too early, then there's always a chance that that patent will publish roughly 18 months after you filed the patent, and then your competitors will have access to see what you've done on your chemistry program. And basically, if the target's interesting enough, they can use your chemistry to start their own chemistry programs and work their way around your patent. So basically, the best objective is to leave it as long as possible before you file your patents around the chemistry. But this depends, of course, on competition. If there are a lot of other companies working on the same target, you might go earlier.
Thanks, Ian.
Thanks. So next up for you, Tom, I've got a question about what are the reasons for basket and umbrella trials being predominantly used in oncology? And then what are some of the key factors behind the low use of these study designs?
Look, I think probably the development of cancer chemotherapeutics has dominated a lot of the drug discovery work, and that's probably part of it. And a lot of the early work for these designs came out of MD Anderson Cancer Center in Texas. So I think that's also partly why as well. But we're certainly seeing now extension to a whole range of areas. So we're doing some work in cystic fibrosis. There are also these platform designs in Alzheimer's and a whole range of areas. So I think that will change. I think it's mostly historic. That the barriers to implementing them, the amount of resources to set this up. So it's a relatively relatively novel. I mean, the statistics that we use, have been around for 20, 30 years or so. A lot of the, we use a lot of computation, and it's computation that we haven't been able to do on desktop computers, except in the past sort of 15 years or so. Before that, a lot of the analyses you just couldn't do. You certainly couldn't do by hand. And I think the clinical trial space is intrinsically fairly conservative and in its approach, with a very high focus on preventing type 1 error. That certainly from a regulatory perspective, that the main priority is not to approve something that doesn't work. And that's pretty well controlled with traditional trial designs. It's just that type 2 error or type 3 error, which is where you never evaluate a drug because you don't have enough patients to evaluate it. They're things that are much better handled, I think, from a Bayesian perspective. So I think it's a mix of things, but I think it will change over the next 10 years or so.
So thank you, everybody. I think we're coming close to where we're out of time, getting close to the hour. So I'd like to thank everybody for their work today. For those answers that we haven't gotten to, we will be able to provide answers to those questions via our website in the coming days. I'd like to thank everybody for joining us. After the webinar, a survey will pop up with a link to the webinar video, and the Q&A will be provided via email, and we look forward to your feedback. I'd like to very much thank Ian and Tom for a fantastic webinar today, to our Q&A facilitators, Chris and Laura, and most importantly to you, the audience, for your active participation. And a very special thank you goes out to the staff of Sydney Children's Hospital Network who have supported today's presentation. Advanced Therapeutics is rapidly evolving and promises to deliver innovative and potentially transformational treatments for children. Please stay tuned for our next webinar, which will be in early December, where we will explore the journey from preclinical research into clinical care. And we look forward to having you join us then. So goodbye for now, and see you all in December. Thank you.
Thank you.